Hello students, welcome to the channel. In today's video, I am going to explain something extremely important for researchers working with non-coding RNA. Okay. So if you are planning to study on non-coding RNA or small non-coding RNA using GEO datasets, you must select the correct type of dataset. If you are selecting wrong dataset, then your entire work will be in soup. So in this video, I will clearly explain what type of RNA sequence you need, how to identify the correct dataset in GEO, and what to avoid and the common mistakes made by the researchers. So first part, let us try to understand what you are trying to look for. So you are looking for non-coding RNA. So there are two major categories here, small non-coding RNA and long non-coding RNA. So within small non-coding RNA, we have miRNA, piRNA, snoRNA, and small nuclear RNAs. So all these four come under small non-coding RNAs. And whereas the other one is the long non-coding RNA. So in this we have lncRNA, circulating RNAs and long intergenic RNAs. So this distinction is extremely important to have a clear knowledge before proceeding further for the downstream processing of the data or while downloading the GEO dataset. Okay. So if you are specifically looking for small non-coding RNA, and if your target is the out of the four small non-coding RNAs, either it is miRNA, piRNAs, snoRNA or other RNAs, so then you need to check a title which is having small RNA sequencing or miRNA sequencing or microRNA sequencing data. Okay, so this is very important. Okay, and when you are looking for long non-coding RNAs, then you need to look for the titles with total RNA sequencing, RNA depletion or ribose 0, strand specific total RNA sequencing. So these terms you need to specifically look in the title so that you will be selecting the right data. Okay, so please avoid the mRNA sequence or bulk RNA sequence, exome sequence, microRNA datasets. Okay, so now let's give a quick search. So suppose if you are interested in, so don't search here. So either you can go for search for studies at GEO datasets. Then you will land into the NCBI GEO dataset homepage. So here you type what is your interest. Suppose you are interested in breast cancer and you type and, okay, to connect two different things. So this is like an advanced search, and then click and then you type miRNA sequence. Okay, then click on search. So now you can see here title, integrating circulating microRNAs with epidemiological factors enhance breast cancer detection across subtype. You can see in brackets they have specifically mentioned miRNA sequence. So these keywords you need to identify specifically in the dataset you are looking for, irrespective of the disease type or cancer type. Okay, and when it is clear, then only you go for the selection. And another important aspect you need to consider here is that whether the data has been just submitted to the database or it has got published. So always it is preferable to select the published data sets. So you can see here in the first one, so this is not linked to any published paper, research article. So it means the data is not published. And in this case, this one, it clearly shows there is link for PubMed, which is a database for literature, and full text in PMC PubMed Central. So it's clearly understandable that this data has been published and we can use this data for our studies. Always go for the published data. Okay. And the another most important thing is that when you are looking at the type, look for non-coding RNA profiling by high throughput sequencing, or at least it should have this word called expression profiling by high throughput sequencing. And always the another thing is that platform is always, it will be GPL, and the number of samples should be greater than 6. Okay, so then you click on the title of the sample, and here you can see need to check for the metadata. So this is the status of the data. It is reported on Feb 18th 2026. Okay, and this is the title of the data submitted, and it belongs to the organism Homo sapiens, and you can see the experiment type, that is by high throughput sequencing, and you can see the summary of the data. And here you can also see the authors who have contributed for this work from here. So from the contributors, so this is all the data related to the authors who have contributed for this work. And if you scroll down, you can see the platforms and all the samples. So this has 12 samples, and you can see all the samples. We have 3 wild type samples and 3 mutation samples, and you can see like there are total of 3 mutations, and for each mutation we have 3 samples. Okay, so it's very clearly indicating that the researchers have worked specifically on the 3 mutations here. So if you are also looking for same kind of mutations, then you can select this data, or else you just have a clear idea what exactly you are searching for, and look for your data and the sample set very carefully. Okay, that fits to your problem or hypothesis that you are aiming for. Okay, and then you can download this data as the raw file using this HTTP. Just click on this HTTP, and you can see that the file will be downloaded as a tar file. So we have to unzip this file, and we are going to use for further analysis. Okay, so thank you guys for listening. To summarize, if you want to miRNA data or small RNA data or lncRNA data, so just look for total RNA sequencing data with rRNA depletion. Okay, so we are not looking for ribosomal RNAs. That is the reason we are mentioning rRNA depletion. So that will also give us the best data. Okay, so if you find this useful, please like, share and subscribe, and me in the next video. I will show you how practically we can filter the GEO datasets for small RNA and long or non-coding RNA datasets. So thank you for watching.